Welcome! The Assistant Professor Biology series presents Cell Division, Mitosis and Cytokinesis. All life arises from pre-existing life through cell division. From the first primitive bacteria in the primordial sea to the multitude of life forms inhabiting the oceans and continents of the Earth today, cell division has propagated all life on our planet. All living things, from the tiniest protozoan to the largest whale, grow, reproduce, and create new body cells through the process of cell division. In this program, we will learn about one type of cell division called mitosis. There are 3 lessons. Lesson 1 explains that mitosis is one of the 3 forms of cell division. Lesson 2 illustrates mitosis and cytokinesis are part of the eukaryotic cell cycle. Lesson 3 details the process of mitosis and cytokinesis. Lesson 1. Mitosis is one type of cell division. There are 2 other types: meiosis and binary fission. Mitosis and meiosis are forms of cell division that occur in eukaryotic cells. With the exception of bacteria, all living organisms, plants, animals, fungi, and protists, are composed of one or more eukaryotic cells. Binary fission is the form of cell division that occurs in prokaryotic cells. Prokaryotic cells are also called bacteria. Let's briefly compare the 3 forms of cell division. Mitosis results in 2 identical cells that have exactly the same genetic information. Meiosis creates special cells called gametes that enable sexual reproduction. These cells contain half the genetic information of the original parent cell. Binary fission, like mitosis, also produces 2 genetically identical cells. Now that we've looked at the 3 types of cell division, let's return to mitosis. Mitosis is how single-celled eukaryotic organisms, like the tiny paramecium shown here, reproduce themselves. Mitosis is also how multicellular organisms grow and develop, from a single-celled fertilized egg into an adult made up of many cells. And mitosis is how new body cells are created, body cells like the red and white blood cells shown here. In multicellular organisms, new body cells are constantly being produced. These new cells replace damaged and worn-out cells and respond to inflammation and infection. Let's review what we've just learned about mitosis and the other 2 types of cell division. Mitosis is one type of cell division. The other 2 types are meiosis and binary fission. Mitosis and meiosis occur in eukaryotic cells. Binary fission occurs in prokaryotic cells. All organisms except bacteria are made up of 1 or more eukaryotic cells. All bacteria are single prokaryotic cells. Mitosis produces 2 genetically identical cells. Meiosis produces cells called gametes that contain 1/2 the genetic information of the original parent cell. Binary fission, like mitosis, also produces 2 genetically identical cells. Mitosis is the process through which single-celled eukaryotic organisms reproduce, multicellular organisms grow and develop, and new body cells are created. Lesson 2. Mitosis is part of the eukaryotic cell cycle. Most eukaryotic cells, whether they are free-living single-celled organisms or the body cells of a plant or animal, undergo a repeated cycle of growth, maintenance, and cell division. This repeated cycle is called the cell cycle. Depending on the type of cell, a cell can go through the entire cycle in just a few minutes, or only once during the entire lifespan of an organism. The cell cycle consists of 5 phases called G1, S, G2, mitosis, and cytokinesis. The first 3 phases, G1, S, and G2, together are called interphase. Interphase is the period of the cell cycle when the cell is not dividing. The last two phases of the cell cycle, mitosis and cytokinesis, together make up the portion of the cell cycle when the cell is actively dividing. During the first part of interphase, the G1 phase, a cell is growing and performing its normal functions. Most cells spend the majority of their lifespan in G1. Many cells actually pause their cell cycle in G1 and enter a resting phase called G0. Cells may remain in G0 for a period ranging from a few days to many years. At any given time, most of the cells of a plant or animal are in G0. Some cells, such as muscle and nerve cells, remain in G0 permanently and never divide. Other cells, such as those that make up the liver, leave G0 and continue their cell cycle only in response to an injury. Once a cell is signaled to divide, it enters the second and third phases of interphase, S and G2. During these phases, the cell prepares for cell division. During S phase, new strands of DNA are synthesized in order to produce a replica copy of the cell's genome. The S phase is complete when the cell has two identical copies of each of its chromosomes. During G2, the duplicated chromosomes begin the process of coiling into tightly compacted packages, and the molecules and organelles required for the cell division process are created. At the end of G2, the cell is ready to enter cell division. Cell division is made up of two division processes. 1. The division of the duplicated chromosomes into two equivalent sets, called mitosis, and 2. The physical division of the cellular cytoplasm to form two new cells, called cytokinesis. Following cytokinesis, each of the two new cells enters into its own cell cycle. Let's review what we've learned about mitosis and how mitosis forms part of the eukaryotic cell cycle. This diagram of a cell cycle describes the life of most eukaryotic cells. Depending upon the cell type, the cell cycle can be as short as a day or as long as many years. The cell cycle consists of 5 phases: G1, S, G2, mitosis, and cytokinesis. The first 3 phases, G1, S, and G2, together comprise interphase, the span of time in the life cycle of a cell when it is not dividing. A cell spends most of its life in the G1 phase, when it is growing and performing its normal functions. Many cells enter a resting stage called G0 and remain there until signaled to divide. Some cells remain in G0 permanently. Preparation for cell division occurs during the last 2 phases of interphase, the S and G2 phases. Duplicate copies of each chromosome are created during the S phase. During the G2 phase, the duplicated chromosomes begin the process of coiling into tightly compacted packages, and molecules and organelles required for the cell division process are created. At the end of G2, the cell is ready to enter mitosis. Cell division represents the portion of the cell cycle when the cell is actively dividing. Mitosis is the process of dividing the duplicated chromosomes. Cytokinesis is the process of dividing the cellular cytoplasm. Following cytokinesis, each of the 2 newly created cells begins its own cell cycle. Lesson 3. Mitosis and cytokinesis. Mitosis is divided into 4 subphases. These phases are named for changes that can easily be distinguished when viewing a dividing cell through a light microscope. Shown here are the 4 phases as they appear in a dividing cell: prophase, metaphase, anaphase, and telophase. This is a cell undergoing mitosis and cytokinesis. The dark strands of material seen in the center of the cell are the duplicated chromosomes. Watch carefully as they are distributed into 2 equivalent sets, followed by the division of the cell's cytoplasm to form two new cells. Now that we've seen the process of mitosis and cytokinesis in an actual cell, let's examine what is happening in more detail. First, let's view an animated sequence of a cell dividing. Watch closely. We will review each step in detail and then watch the entire sequence again. The first phase of mitosis is prophase, the longest of the four phases. Four key events happen during this phase. The first event marks the beginning of mitosis and prophase. During this event, the chromosome condensation that began during G2 reaches the point where the duplicated chromosomes become so compact that they are visible with a light microscope. Each compact chromosome consists of two identical copies attached at a central point. The two copies are called sister chromatids, and the attachment point is called the centromere. The second key event of prophase is the assembly of the mitotic spindle apparatus. In animal cells, two pairs of organelles called centrioles move to opposite poles of the cell, forming the fibers of the spindle apparatus between them as they move. Plant cells do not have centrioles, but do form a similar spindle apparatus. The spindle apparatus controls the orderly separation of the duplicated chromosomes into two equal sets. In animal cells, an array of fibers called the aster is formed when the centrioles reach the opposite poles. The aster is thought to help brace the mitotic spindle apparatus as it moves the duplicated chromosomes. Plant cells, which have rigid walls, do not form asters. The third major event in prophase is the disintegration of the nuclear envelope. The fourth important event in prophase is the growth of another group of spindle fibers. These fibers grow toward the chromosomes through the disintegrating nuclear envelope. Watch the animation of prophase again, paying close attention to the four key events. The second phase of mitosis is metaphase. Two key events happen during metaphase. The first event, which marks the beginning of metaphase, is the attachment of some of the spindle fibers to the centromeres of the chromosomes. This attachment occurs at special sites called kinetochores. The fibers attach to opposite sides of each centromere, one attachment site for each sister chromatid. The second key event in metaphase is the movement of the chromosomes toward the center of the cell, where they are positioned in a circle along a central plane. Each chromosome is positioned along the plane with its centromere in the middle and sister chromatids on either side. Watch the animation of metaphase again, paying close attention to the two major events. The third and shortest phase of mitosis is anaphase. Three important events occur in anaphase. The first event, the one that marks the beginning of anaphase, is the division of the chromosome centromeres, separating the sister chromatids. Once this division has occurred, each sister chromatid is once again referred to as a chromosome. The second event is the shortening of the attached spindle fibers, which moves the chromosome copies to opposite poles of the cell. The third event is the lengthening of other fibers of the spindle apparatus, an action that elongates the cell. Anaphase is complete when the opposite poles of the dividing cell contain equivalent collections of chromosomes. Watch the animation of anaphase again, paying close attention to the three events. Telophase is the final phase of mitosis and is roughly the opposite of prophase. Three important events occur during telophase. All three events of telophase proceed simultaneously. The mitotic spindle apparatus is disassembled, a nuclear envelope reforms around each set of chromosomes, and the chromosomes unwind from the tightly packed form. At the end of telophase, there are two equivalent sets of chromosomes at opposite poles of the cell. Mitosis is complete at the end of telophase, when the cell has portioned its replicated genome into two nuclei at opposite poles of the cell. Watch the animation of telophase again, paying close attention to the three key events. The final physical division of the cellular cytoplasm is a process called cytokinesis. Cytokinesis actually begins during the latter part of anaphase and continues beyond the end of telophase until the cell is completely divided. In animal cells, cytokinesis occurs through a process called cleavage. Cleavage begins with the appearance of a groove that rings the cell. This groove is called the cleavage furrow. The furrow is created by the tightening of a ring of fibers attached to the internal surface of the cell. The fibers contract, pulling the surface of the cell together like a drawstring until the cell is completely divided. Cytokinesis is a different process for plant cells. Because they are enclosed in a rigid cell wall, plant cells do not pinch in two like an animal cell. A dividing plant cell forms a partition called the cell plate between the two new nuclei. The plate is formed from cell membrane vesicles that line up along a central plane. This animation shows how vesicles fuse together to form the cell plate. The cell plate grows outward until it reaches and fuses with the inner cell membrane, effectively dividing the cell in two. Cellulose is then laid down, forming two new cell walls. Let's review what we've learned about mitosis and cytokinesis. Mitosis is the cell division process that ensures the equal distribution of duplicate sets of chromosomes into two new daughter cells. Mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. Prophase is the longest phase of mitosis and encompasses four key events: the completion of chromosome condensation, the formation of the mitotic spindle apparatus. Near the end of prophase, the last two events occur. The nuclear envelope disintegrates, and fibers of the spindle apparatus grow in toward the chromosomes. The second phase of mitosis is metaphase. Metaphase begins when fibers of the spindle apparatus attach to the centromeres of the chromosomes. The fibers move the chromosomes toward the center of the cell, positioning them so that the centromeres of each chromosome are aligned with the sister chromatids on either side. Anaphase is the third and shortest phase of mitosis. The first of three key events that occur in anaphase is the simultaneous division of the centromeres of all the chromosomes, separating the sister chromatids. Following separation, the sister chromatids are moved by the spindle fibers to opposite poles of the cell. At the same time, other fibers of the spindle apparatus lengthen, elongating the cell. Anaphase is complete when equivalent collections of chromosomes are positioned at the opposite poles of the dividing cell. The fourth and final phase of mitosis is telophase. During telophase, three key events proceed simultaneously. The spindle apparatus is disassembled, a nuclear envelope forms around each set of chromosomes, and the chromosomes unwind from their condensed form. Cytokinesis is the process that completes the physical division of the cell. Animal cells undergo cleavage, a process similar to the pulling together of a drawstring. In contrast, plant cells construct a central cell plate from the fusion of tiny vesicles. This central plate ultimately forms the membranes and cell walls that separate the new cells. Now that we have reviewed each phase of mitosis, prophase, metaphase, anaphase, and telophase, and the process of cytokinesis, let's put it all together and see the complete sequence again. Thank you for selecting this program from Allied Video's Assistant Professor Biology series. For more information on other Allied Video products, please call 1-800-926-5892 or visit our website at www.alliedvideo.com.